Before entering into the laboratory, make sure to use personal protective equipment such as handscoon, nurse cap, face mask, and laboratory coat. The second step that must be done is to sterilize the tools that will be used. The tools that were sterilized with an oven were 6 Petri dishes, 1 mortar pestle, 1 10 ml measuring pipette. Bake in the oven at 180 degrees for 1 hour. And as for the tools that were sterilized by autoclaving is 5 test tubes with 9 ml of NaCl, blue tips, Erlenmeyer with 90 ml distilled water. Autoclave at 121 degrees for 15 minutes. The first step is making media with nutrient agar. Prepare 1 Erlenmeyer 250 ml, then add 150 ml of distilled water for 6 Petri dishes. Weigh 4.2 until 4.5 grams of nutrient agar medium and add it to the Erlenmeyer later. Dissolve the media liquid with a hot plate at 150 degrees Celsius for approximately 5 until 7 minutes. Let it boil, then proceed by autoclaving the nutrient agar medium at 121 degrees for 15 minutes. In our examination of the germ count, we use solid food samples, namely the traditional something snack. The preparation of food specimens is carried out in the BC to avoid contamination. As for the steps in making the first food sample, the specimen is crushed using a pestle mortar, then weigh the specimen weighing 10 grams. Second, enter 10 grams of material into the Erlenmeyer flask which already contains 90 ml of sterile aqueous. Third, shake about 25 times until homogeneous. Next steps in carrying out an examination of the germ count. First, prepare 1 Erlenmeyer containing 90 ml of distilled water which has been sterilized for the first dilution and 5 test tubes containing 9 ml of distilled water each for dilutions 2 to 6. Next, prepare 7 sterile Petri dishes and mark the 6 Petri dishes according to the code, dilution and inspection date. Then take 10 grams of the specimen and put it in the Erlenmeyer, then homogenize it. Then take 1 ml of material from the Erlenmeyer and put it in 2 tubes with a micropipette. The liquid is homogenized with a vortex mixer, and so on until the 6th tube. Take 1 ml, then put it into each Petri dish according to the same dilution code, starting with the 6th tube using a sterile pipette. Then pour the liquid nutrient agar that has been prepared into each Petri dish. Next, the Petri dish is shaken slowly to form number 8 until it is homogeneous and left to cool and freeze. Also make 1 media that is not added to the sample as a control medium. Then put it in the incubator 37 off for 2x 24 hours upside down. After being in the incubator with a temperature of 37 degrees for 2 times 24 hours, the media is ready to be observed. Count the number of colonies that grew on the Petri dish and then report the results of the colony count. The reading of the results is carried out by calculating only carried out on a Petri dish that produces a colony count of between 30 until 300, and if the number on a control Petri dish is more than 10, the number of colonies on each Petri dish must first be reduced by the number of colonies on the control Petri dish. Conclusion. Based on the practicum examination of germ counts in something traditional food samples with the total plate number method, the results obtained were that p-value of bacterial colonies in the food test samples chips was 1,825 colony forming units per gram. According to the provisions of the Indonesian Food and Drug Supervisory Agency, that the requirement or standard for the odd value in food is 1x10⁶ colony forming unit per gram.
Clean the BST area that has been used. After that, wash the tools that have been used. Don't forget to removing disposable personal protective equipment such as handscoon and nurse cap. And for the last, don't forget to wash your hands.